Breakdown alone cannot sustain life. At some point, what has been dismantled must be rebuilt.
A simple but profound truth.
Welcome back to the deep dive. We are shifting gears today in a really major way.
We are.
For the last few weeks, if you've been following along with our biochemistry series, we have been absolutely obsessed with destruction.
It has been a bit violent, hasn't it? A lot of smashing and burning.
It really has. We've been deep in catabolism, smashing glucose molecules, breaking down fats, ripping carbon chains apart just to squeeze out that precious ATP.
All for the energy harvest.
Exactly. It's been entirely about generating power. But Today we flipped the script. We are entering the era of what I'm calling the Great Rebuild.
Anabolism.
Precisely. Anabolism. This is the other side of the coin. This is how your body takes a bunch of simple spare parts, some carbon skeletons from your breakfast, a little bit of nitrogen from the air, indirectly, and somehow builds a complex human being.
It's the construction phase.
And let me tell you, after digging into the reading for this one, specifically trying to synthesize the systems logic from Leninger principles of biochemistry, and then grounding it with the clinical realities from Harper's Illustrated Biochemistry, I have a completely new respect for the sheer logistics happening inside my cells right this second.
It is impressive engineering.
Yeah.
But it's also a completely different economic model from what we've been looking at.
That's the metaphor I want to start with. We used to compare catabolism to, say, a furnace just burning fuel for heat.
Right. Exergonic. Releasing energy.
But anabolism is like a very, very conservative, maybe even grumpy master cracksman.
Grumpy, I like that. Why grumpy?
Well, extremely intolerant of waste. I mean, truly, the cell never builds what it doesn't need. It recycles absolutely everything it can. And the blueprints, they are so fiercely regulated.
You don't just start building.
No, you don't just decide to build a nucleotide because you feel like it. You need a permit, signed in triplicate, probably approved by 3 different committees.
That is a very, very accurate way to frame it. Biosynthesis is expensive. I mean, energetically speaking, it costs a fortune in ATP and in reducing power. So the body has evolved to be a total miser.
So if catabolism is about generating wealth in the form of energy,
Anabolism is about spending it. And you have to spend it wisely. There are no frivolous purchases in the cell's economy.
So today, our mission is to explore this expensive side of metabolism. We are going to look at how we synthesize amino acids, the building blocks of protein and nucleotides, the building blocks of your DNA and RNA.
The machines and the code.
Exactly. And we're going to tackle a central question that just kept bothering me while I was reading. If our bodies are so smart, why can we manufacture some amino acids from scratch, but we absolutely have to eat others?
The paradox of the essential versus the non-essential.
Right. It feels like a design flaw on the surface. I can make alanine no problem at the drop of a hat, but tryptophan, I have to go find a turkey sandwich, or eventually I'm in serious trouble. Why did we lose that code?
It's a fascinating evolutionary trade-off. But, before we get to the specifics of the amino acids, we really need to lay down the ground rules of the factory.
Okay.
We mentioned that anabolism is expensive. In biochemical terms, we say it is endergonic.
Which just means it requires an energy input. It's an uphill reaction. You have to push the boulder up the hill.
Correct. But it's also reductive. And this is a key distinction that Leninger makes very, very clearly.
Okay, reductive versus oxidative.
Right. Catabolism is oxidative. We strip electrons off the fuel to make energy, and we store those electrons, usually as NADH.
Yep, feeding the electron transport chain.
Anabolism is the exact opposite. We use electron donors to build complex molecules up from simple ones. We're adding electrons back in. And usually, the electron donor isn't NADH.
It's NADPH.
Exactly. The phosphorylated version. It seems like such a tiny change, just one phosphate group, but it's like a tag. It tells a cell, these electrons are for building, not for burning.
It's like having 2 different bank accounts. NADH is your checking account for paying the daily bills and keeping the lights on at the mitochondrial power plant.
Yeah, that works.
And NADPH is your dedicated savings account, strictly for, home renovations and building new additions to the house. You don't mix the funds.
That's a perfect analogy.
Yeah.
It's metabolic compartmentalization, but with currency instead of location. So anabolism is about fighting entropy. You are taking disordered, simple molecules and forcing them into these complex, ordered structures. And that fight requires resources.
Okay, so with those rules in place, endergonic and reductive, let's go back to my question. Why can't we make everything? Harper lays out the amino acids into nutritionally essential and nutritionally non-essential.
And Strictly speaking, Harper lists 8 that are absolutely essential, meaning we have zero capacity to synthesize them. Others are what we call conditionally essential, which we can get to.
Right. But just looking at the list, have you noticed a pattern in which ones we kept and which ones we lost the ability to make?
I did, yeah. It seems pretty obvious once you look at the molecules.
Yeah.
The ones we can make, like alanine, glutamate, aspartate, they seem simple. They're small.
Very simple structures.
But those we have to eat, tryptophan, phenylalanine, histidine, those are huge, complex molecules. They're these big aromatic rings, complicated side chains.
Precisely. And Harper suggests a concept for this that I just love. It's called negative survival value.
Negative survival value. It sounds so counterintuitive.
It blew my mind. It's basically the idea that knowing how to do something or having the machinery for it can actually hurt you in the long run.
Okay, unpack that. How does that work?
Think about it from an evolutionary perspective.
Yeah.
If you are an early human ancestor, living in an environment where you are eating plenty of plants or other animals, you are getting a steady supply of tryptophan from your diet. It's abundant.
Right.
Now, tryptophan is, as we said, incredibly hard to build. The pathway is something like 15 steps long. It takes dozens of enzymes, a massive amount of ATP, and a long, long stretch of DNA to code for it all.
So maintaining that entire factory line is a huge metabolic burden.
It's dead weight. It's like paying for a factory you never use. So imagine a mutation happens that breaks your tryptophan factory.
Okay.
As long as you're eating tryptophan every day, you don't die. In fact, you're now at an advantage. You save energy. You no longer have to build those 15 enzymes. You are leaner and more efficient than your neighbor who is still wasting ATP and resources building it from scratch.
So the lazy mutant wins the evolutionary race.
In an environment of abundance, yes, we effectively outsourced the hard work. We looked at plants and bacteria and said, you guys handle the heavy, complicated chemistry, we'll just eat you.
That is evolutionary downsizing at its finest. It's incredible.
It makes you realize that our DNA isn't just a list of what we can do, it's also a history of what we decided we didn't need to do anymore.
And that strategy works perfectly, right up until the moment that food source disappears.
Which is when you get deficiency diseases.
Right.
So that explains the essential ones. Yeah, let's talk about the ones we did keep, the non-essentials.
The ones we make ourselves.
I was dreading this part of the reading because I thought, oh no, here we go, 20 different disconnected pathways to memorize. But Leninger organizes them in such a helpful, logical way.
The biosynthetic families.
Yes. Instead of treating them as 20 isolated chemicals, you just group them by their parent molecule. And the amazing thing is, the parents are all old friends of ours.
They are. They all come from the 3 central metabolic pathways we've already spent weeks on. Glycolysis, the citric acid cycle, or the pentose phosphate pathway.
It makes the map so much smaller and more manageable. You have the alpha-ketoglutarate family, the pyruvate family, the 3-phosphoglycerate family. It's like a metabolic reunion.
It really emphasizes that metabolism isn't a series of separate silos. It's a deeply interconnected network. You can just pull a carbon skeleton out of the Krebs cycle, say alpha-ketoglutarate, and with a few tweaks, it becomes an amino acid.
Okay, but here's the big hurdle. Carbon skeletons are fine. We have plenty of carbon from sugar and fat breakdown. But amino acids, by definition, need an amine group. They need nitrogen.
And nitrogen is tricky.
It seems like it's the limiting region for all of life.
In many contexts, it is. And chemically, free ammonia is very toxic to us, especially to the brain. You can't just have free ammonia floating around your cells. It disrupts pH, it messes with neurotransmission. It's just bad news.
So we need a handler. We need a fixer, a designated molecule that can grab onto the nitrogen and hold it safely until it's needed.
And we have 2 main fixers. The amino acids, glutamate and glutamine. These are the molecules that interface with the raw inorganic nitrogen.
I started picturing this as a kind of funnel. All the nitrogen that's going to be used for building things in the body has to first pass through this glutamate and glutamine port.
That's a great visualization. And the key enzyme here, the gatekeeper of the funnel, is glutamine synthetase. Leninger really highlights this as a major regulatory checkpoint.
Because it's the entry port.
It's the main entry port. It takes glutamate, which already has 1 nitrogen, and it uses ATP to force another ammonium ion onto it to make glutamine. This is the primary way our body mops up toxic free ammonia and prepares it for use in biosynthesis.
And once you have that nitrogen safely attached to an amino acid, you can start swapping it around.
Exactly.
This is that transamination concept we touched on in catabolism, but now we're using it to build things, not break them down.
Correct. It's like modular construction.
Yeah.
You have an amino acid with a nitrogen group, And a keto acid, which is just a carbon skeleton without one, you use an enzyme, a transaminase, to just swap the amine group over. You click the nitrogen onto a new carbon skeleton, and voila, you have a new amino acid.
It's so efficient. So let's walk through the families using that logic. Family number 1, the alpha-ketoglutarate family.
Okay.
This is the glutamate family.
Right. Alpha-ketoglutarate is a 5-carbon intermediate from the citric acid cycle.
We've seen it 100 times.
You add a nitrogen to it via transamination, and you get glutamate. It's a one-step conversion. Very simple. But then glutamate becomes the parent for the others in the family. Glutamine, proline, and arginine.
Proline was really interesting to me because it's got such a weird shape. It's that rigid ring structure that puts a kink in protein chains.
And if you look at its structure, proline is essentially glutamate biting its own tail. The chemistry is a bit more complex, but it involves reducing one of the carboxyl groups to an aldehyde, which then spontaneously reacts with the amine group to close the ring.
It cyclizes itself.
Exactly. It's a cyclized version of glutamate. Arginine is more complex, part of the urea cycle, but it also derives from this same family.
Okay, that's family 1. Then we have the 3-phosphoglycerate family. 3-PG comes straight out of glycolysis.
This is the parent for serine, a 3-carbon amino acid. And serine is a very generous molecule. It's a key donor in 1-carbon metabolism.
What does that mean, a 1-carbon metabolism?
It's a whole system for moving single carbon atoms around for synthesis. And serine can give up one of its carbons to become glycine, which is the simplest amino acid, just 2 carbons.
So serine to glycine is a donation?
A donation of a carbon atom. And that carbon atom, by the way, is carried by our friend tetrahydrofolate, which we'll see again later.
Always comes back to folate. Now, in this same family is cysteine. And this is where Harper threw in a curveball that I think is really important for people to understand. Cysteine is technically listed as non-essential. We can make it, but there's a big catch.
There is the sulfur. Cysteine is one of 2 amino acids that contains a sulfur atom. And our bodies cannot manufacture sulfur atoms. We are not, nuclear reactors.
Right. We have to ingest it.
So we make the carbon backbone of cysteine from serine, but we have to steal the sulfur atom from another amino acid, methionine.
And methionine is an essential amino acid. We have to eat it.
Exactly. So if you don't eat enough methionine in your diet, you physically cannot make cysteine. Your cysteine factory has no raw materials for the sulfur part.
So that's what conditionally essential means.
Precisely. We can build the car, but only if you bring us the steel from the outside. If the steel supply runs out, the factory shuts down.
It really blurs that line between what we can and can't do. It's all dependent on the availability of raw materials.
Which brings us to the last couple of simple families, the pyruvate and oxaloacetate families.
These seem the most straightforward of all.
They are. They're just direct transaminations. You take pyruvate, the end product of glycolysis.
Add a nitrogen.
And you get alanine. Next step, done. You take oxaloacetate from the Krebs cycle.
Add a nitrogen.
And you get aspartate. Again, one step. And then aspartate can be modified to become asparagine. And fascinatingly, the enzyme that makes asparagine from aspartate uses glutamine as the nitrogen donor.
So glutamine, our nitrogen handler, passes the nitrogen hot potato over to aspartate to make asparagine. It really is a funnel.
It is. The whole system is designed to move nitrogen safely and efficiently from the entry point to wherever it's needed. But, listing all these pathways is the happy path. That's when the factory is running smoothly.
And all the parts are there and the machines are working?
Right. To really understand how critical this all is, we have to look at what happens when the manufacturing plant has a bad day.
Or when a single specific tool is missing from the toolbox. This is where Harper just shines, connecting the lines on the map to a patient in a hospital bed.
Absolutely.
Let's start with a classic thought experiment, or rather, a historical reality. The sailor's scurvy. We all know the story: sailors on long voyages, no fresh fruit, their gums start bleeding, their teeth fall out, old wounds reopen.
A truly horrific condition.
And we know it's vitamin C deficiency. But biologically, what is actually breaking? Because I always had this vague idea that vitamin C was just, generally good for your immune system.
It's so much more mechanical and specific than that. Scurvy is a failure of anabolic post-processing.
Okay, post-processing, what does that mean?
It means after the initial synthesis. So your body builds the primary protein chain for collagen using regular proline and lysine. But collagen is unique. It needs to be incredibly strong. It's the connective tissue, the rebar, holding your entire body together.
It's the rope that ties our cells into a solid functional shape.
Exactly. And to make that rope strong, the body adds hydroxyl groups, OH groups, to many of the proline and lysine residues after the protein has already been built. This is called a post-translational modification.
So you make proline, you put it into the protein chain, and then you come back and modify it to hydroxyproline.
Yes. And that hydroxyproline is critical. It allows the individual collagen fibers to form strong hydrogen bonds with each other, letting them twist tightly into a superstable triple helix. It acts like molecular Velcro.
And that's what gives it its strength.
It raises the melting point of the collagen significantly.
The melting point, that is such a visceral and terrifying image. Without vitamin C, your collagen literally melts at body temperature.
It becomes unstable at body temperature, yes. If you don't have those hydroxyl groups, the collagen triple helix can't form properly. Now, the enzyme that does this hydroxylation is called prolyl hydroxylase, needs iron to work.
Okay.
But the iron has to be in a specific reduced state, the F2 plus state. During the reaction, it gets oxidized to F3 plus SD, which inactivates the enzyme.
So the tool gets rusty after one use?
A perfect way to put it.
Yeah.
And vitamin C ascorbate is the mechanic that comes along and reduces the iron back to its working Ft plus state. It polishes the rust off the tool.
So no vitamin C means the iron gets stuck in the oxidized state, the enzyme stops working, you can't make hydroxyproline, the collagen can't form stable cross-link.
And your connective tissue literally loses its structural integrity. Old wound scars, which are made of collagen, break down and reopen. Gums bleed because the capillaries lose their strength and just rupture under normal blood pressure.
Yeah.
Joints swell. You are literally unraveling because the glue holding you together has failed.
That's terrifying. And it's all because of one little cofactor for one specific anabolic enzyme.
That's the fragility of these complex systems. Let's look at another one, a genetic defect this time, not dietary. PKU, or phenylketonuria.
This one ties right back to that essential versus non-essential trade-off we started with.
It does. Phenylalanine is essential. We have to eat it. Tyrosine is considered non-essential. We can make it.
But the catch is how we make tyrosine. We make it from phenylalanine. There is a single enzyme, phenylalanine hydroxylase, that converts the essential Phe into the non-essential tier. It just adds one OH group.
So phenylalanine is the direct precursor, the raw material for tyrosine.
Exactly. But in individuals with PKU, that converter enzyme is broken. It's genetically defective.
So you get a massive metabolic traffic jam.
A really dangerous traffic jam. Phenylalanine from the diet builds up to incredibly high levels, but since it can't go down the main highway to become tyrosine, it starts spilling over into these little side roads.
It gets converted into other things.
Right. It turns into phenylpyruvate and other so-called phenylketones. And these compounds at high concentrations are neurotoxic. They severely damage the developing brain, causing profound intellectual disability if left untreated.
But there's a double whammy here, isn't there? It's not just the toxicity.
Exactly. That's the other side of the coin. At the same time that phenylalanine is building up to toxic levels, you have a critical shortage of tyrosine because you can't make it anymore.
So for a patient with PKU, tyrosine suddenly becomes an essential amino acid.
Correct. The entire management of the disease is built on this fact. They have to eat a diet with special medical food that is very low in phenylalanine but supplemented with high amounts of tyrosine.
It perfectly illustrates how essential isn't a fixed universal rule. It's a fluid concept that depends on your personal genetics and your health. One person's non-essential nutrient is another person's vital medicine.
That's a huge insight. It makes you realize that dietary advice can never be one size fits all. It's so heavily dependent on your personal enzymatic machinery.
Absolutely. The blueprint you're born with.
Yeah. Okay, let's leave amino acids for a moment and move to the other side of the anabolic house, the synthesis of nucleotides. The A's, G's, C's, and T's.
The blueprints and the currency.
Right, I'm glad you said that because we can't forget that APP is a nucleotide too. It's not just for making DNA, it's the fundamental energy battery of the cell.
Correct. Now, synthesizing nucleotides is arguably even more expensive and complex than amino acids. And just like with amino acids, the cell has evolved strategies to manage that incredible cost.
Two main strategies, right? De novo and salvage.
Exactly. De novo means from new, or making it from scratch, out of very simple precursors like amino acids and CO2.
Artisanal from the ground up.
Yeah, and it's very energy intensive. And then there's the salvage pathway, which is exactly what it sounds like. It's recycling used parts, grabbing pre-made bases and nucleosides from the breakdown of old DNA and RNA and reusing them.
Let's look at de novo first. The difference in the strategy between making purines, that's adenine and guanine, and pyrimidines, cytosine, uracil, and thymine, is fascinating. It's like two completely different architectural philosophies.
It really is. Let's start with purines. The purine ring structure is the more complex one. It has two rings fused together.
Right.
So you might think, logically, that the body would build that complex ring structure separately and then once it's complete, attach it to the sugar backbone.
That would make sense to me. Build the engine, then put it in the car.
But for purines, it does the exact opposite.
Yeah.
Starts with the car chassis, the sugar, specifically a molecule called PRPP, which is phosphoryl pyrophosphate, and it builds the engine piece by piece directly on top of the chassis.
I called this the Frankenstein assembly in my notes. Because it doesn't build it from one precursor, it grabs a nitrogen from glutamine here, a few atoms from glycine there.
It's exactly like that. It stitches them together. The final ring is a mosaic built from glycine, aspartate, glutamine, CO2, and even 1 carbon units carried by tetrahydrofolate, atom by atom, right onto the sugar.
And Leininger really emphasizes the cost. It takes at least 5 ATPs just to build the parent molecule before you even specify it as A or G.
Yes, to get to the first complete purine nucleotide, which is IMP anisinate. IMP is the common ancestor, the parent purine. From IMP, the pathway then branches off to make either AMP for adenine or GMP for guanine.
Okay, so that's the purine strategy. Build it on the sugar. What about pyrimidines?
They flip the strategy completely. For pyrimidines, which have a simpler single-ring structure, the ring is built first, floating free in the cytosol. This is the orotate ring.
So they do build the engine first.
They build the engine first, and only after that orotate ring is fully assembled is it then attached to the sugar chassis, the PRPP.
That's amazing. Two totally different approaches for the same ultimate goal.
And there is a crucial detail here that Harper points out regarding compartmentalization. The very first step of pyrimidine synthesis involves making a molecule called carbamoyl phosphate.
No, wait, that sounds familiar. Carbamoyl phosphate is also used in the urea cycle to get rid of nitrogen waste, right?
Exactly. So the cell has a problem. How does it know if the carbamoyl phosphate it's making is destined for peeing out nitrogen via the urea cycle or for building DNA via pyrimidines?
You don't want to mix those up.
You really don't. And the solution is zoning laws.
Yeah.
Physical separation. The urea cycle happens in the mitochondria. That uses an enzyme called carbamoyl phosphate synthetase I, or CPSI.
Okay.
Pyridine synthesis happens out in the cytosol. That uses a completely different, separate enzyme called CPS2.
I love that. Physical separation prevents metabolic crosstalk. It's like having the kitchen in a different building from the bathroom to make sure the pipes don't get crossed.
And we see what happens when the pipes do get crossed, or when the assembly line fails. There is a rare genetic condition called orotic aciduria.
This is where the pyrimidine ring, the orotate, is built correctly, but you can't attach it to the sugar.
Precisely. The enzyme that connects the finish ring to the PRPP sugar, an enzyme called orotate phosphorubylosyltransferase, is defective. So orotate, the ring, just builds up and spills out into the urine.
And the patient is starved for pyrimidines.
Right. They can't make DNA or RNA efficiently. So they present with growth retardation, developmental delay, and a severe megaloblastic anemia.
And the treatment is so simple, it's beautiful, you just feed them uridine.
You bypass the block. Uridine is a nucleoside. It's a pyrimidine base that already has the sugar attached. The cell can pick that up and use the salvage pathways to phosphorylate it and turn it into the nucleotides it needs. It's elegant biochemical hacking.
Speaking of hacking, the regulation of all of this is just mind-bendingly complex. You can't just make a million adenines and no guanines. The ratios matter tremendously.
They matter immensely. For DNA, you need base pairs. A pairs with T, G pairs with C. If you have a cellular pool that's 90 per G and only 10% A, your DNA replication is going to stall, and the error rate will skyrocket. The cell has to maintain a balanced supply.
And Leininger describes this beautiful system of concerted inhibition and reciprocal control. It's not just one thing stopping the factory, it's everything talking to everything else.
Let's look at that branch point from IMP. Remember, IMP is the common parent of both AMP and GMP.
Right. It's the fork in the road.
To make AMP from IMP, the cell uses GTP as the energy source.
Wait. To make adenine, it burns guanine.
Yes. And to make GMP, the guanine nucleotide, it uses ATP as the energy source.
That is brilliant. It's a reciprocal check and balance system.
Exactly. If you have lots and lots of ATP, that high concentration drives the synthesis of GMP. If you have lots of GTP, that drives the synthesis of AMP. This cross-dependency forces the two pools to stay balanced. It prevents one from running away from the other.
It's like biochemical socialism, from each according to his ability to each according to his need.
In a way, yes. And of course, there's classic feedback in addition. If you have too much AMP at the end of the line, it goes all the way back and inhibits one of the first enzymes in the pathway, shutting off the tap.
It's all about homeostasis. But what happens when the recycling plant breaks? We talked about those salvage pathways, the energy-efficient way of reusing old nucleotides.
Right.
This brought us to what was, for me, the most disturbing disease in the entire chapter, Lesch-Nyhan syndrome.
This is a defect in the salvage of purines. The specific enzyme is HGPRT. It's a mouthful. Hypoxanthine guanine phosphoribosyltransferase.
I practiced that one for a while. So normally, free purine bases like hypoxanthine and guanine, which come from the breakdown of old DNA and RNA, are grabbed by this HGPRT enzyme.
And it sticks them back onto a purine pre-P sugar to be reused as fresh nucleotides.
It saves a huge amount of energy, and it keeps the levels of purine breakdown products low.
Yeah.
But in Lesch-Nyhan, this enzyme is genetically absent.
The recycling center is closed permanently.
And the symptoms are just devastating. Harper lists severe neurological issues, hyperuricemia leading to gout, and the most specific and tragic symptom, compulsive self-mutilation.
Yes. Patients who are typically young boys, because it's an X-linked disorder, will uncontrollably bite their lips, their tongues, their fingers, often to the point of amputation. It is a compulsion, not a lack of sensation. They feel the pain, but they cannot stop.
Why? I mean, what is the biochemical link? Why does a missing recycling enzyme cause the brain to want to destroy the body?
It's one of the great tragic mysteries in biochemistry, but the leading theory connects it to the brain's unique metabolism. The brain, more than many other tissues, doesn't like doing that expensive de novo synthesis. It relies very heavily on recycling, on the salvage pathway.
So when that fails, the brain is in trouble.
It's in big trouble. And what happens is the PRPP levels in the cell skyrocket because PRPP is not being used up by the salvage enzyme.
And all that excess PRPP acts as a powerful signal to ramp up the de novo factory.
Exactly. The cell sees all this available PRPP and thinks, wow, we must really need to build purines from scratch. So the de novo pathway goes into overdrive.
But the cell doesn't actually need all those purines.
No, so they just get broken down into the waste product, which is uric acid. This causes the hyperuricemia and the gout-like symptoms. But the neurological aspect suggests that these purine pathways are also critical for signaling in the brain, perhaps regulating dopamine pathways in the basal ganglia.
So it's a metabolic error that manifests as a behavioral disorder.
A single enzyme error, a single missing protein, and the result is one of the most tragic behavioral disorders known. It is humbling.
It is. Let's touch on gout itself quickly, since you mentioned it. This is the so-called King's disease.
Historically, yeah. It was associated with rich diets, lots of red meat and alcohol, which are high in cells and therefore high in purines. Gout is caused by high levels of uric acid in the blood.
And uric acid is the end of the line for purine trash in humans.
For us, yes. But it's interesting. Most other mammals have an enzyme called uricase that breaks uric acid down one step further to a compound called allantoin, which is very soluble in water. They just pee it out, no problem.
But humans and other great apes, we lost the gene for uricase during our evolution.
So we are stuck with uric acid. The problem is uric acid is not very soluble in water. If the concentration gets too high, either from overproduction or under excretion by the kidneys, it precipitates. It turns into sharp needle-like crystals of sodium urate.
And for some reason, it loves to crystallize in the cooler parts of the body.
Like the big toe.
The classic gout presentation, an excruciatingly painful, red, swollen big toe.
The crystals form in the synovial fluid of the joint. The immune system sees these crystals as foreign invaders and launches a massive inflammatory attack against them. Patients say it feels like having ground glass being churned inside your joint.
And this connects to other diseases too, right? Like von Gierke disease, which is a glycogen storage disease.
Right. That's a perfect example of the metabolic web. Von Gierke is a deficiency in glucose-6-phosphatase. This traps glucose inside liver cells. The glucose has nowhere to go, so a lot of it gets shunted into the pentose-phospho pathway,
Which produces a ton of ribose 5-phosphate.
which is the precursor for PRBP,
Which drives de novo purine synthesis into overdrive.
which leads to a massive overproduction of uric acid and severe gout, even in children. It's all connected. The sugar metabolism pathway crashes directly into the nucleotide metabolism pathway.
The metabolic web. You tug on one string over here, sugar, and the whole web vibrates, knocking the purines out of balance over there.
You've got it.
Okay, final section. We've built the nucleotides. We have our pool of A's, G's, C's, and us. But wait, we built them as ribonucleotides? That's RNA. We need DNA for replication.
Correct. The precursors are always synthesized as ribonucleotides first. ADP, GDP, CDP, UDP. To make the building blocks for DNA, we have to perform one crucial modification. Remove the oxygen atom from the 2' position of the ribose ring to make it deoxyribose.
And this brings us to one of my new favorite enzymes, ribonucleotide reductase, or RNR.
The gatekeeper. This single enzyme is responsible for creating all the building blocks of DNA. It is absolutely essential for life.
And the mechanism is just wild. Leninger describes a free radical mechanism. Usually, we are taught that free radicals are bad, they're reactive oxygen species, they destroy tissue.
They're highly reactive and dangerous. But RNR is an example of an enzyme that has learned to harness that power. It generates and stabilizes a free radical, specifically on a tyrosine residue within the enzyme, and uses it to do this very difficult chemistry. It's like using a controlled explosion to weld something. The enzyme holds the radical very, very carefully to pluck that specific oxygen atom off the ribose ring without blowing up the rest of the molecule.
That's incredible. And the regulation of RNR is maybe the most complex and elegant of all. It has 2 different regulatory switches.
It has to, because RNR controls the raw material for DNA replication. If you get the balance of the 4 DNTPs wrong, say too much DATP and not enough DCTP, you get mutations. The DNA polymerase will make mistakes. So switch number 1 is the activity site. This is the main on-off switch.
ATP in the site turns it on and DATP turns it off.
Right. If you have plenty of energy, ATP, the cell is cleared to make DNA. But if the end product, DATP, builds up, it signals that the DNA precursor pool is full and the enzyme shuts down. Simple negative feedback.
But switch number 2 is the specificity site.
This is the truly cool part. This site tells the enzyme which of the 4 bases to work on next.
Because it's the same enzyme processing all 4.
Exactly. You don't want it to just churn out thymine all day long. So, for example, binding ATP at the specificity site tells the enzyme, make pyrimides, CDP and UDP. Once the pyrimidine products build up, they bind to the site and tell it, okay, we have enough of those, now make GDP. It constantly rotates its own production schedule based on what's currently in the waiting room.
To ensure a perfectly balanced pool for high-fidelity replication.
Precisely. And because RNR is so absolutely critical for making DNA, it is a prime target for cancer therapy.
That's the big clinical application here. Chemotherapy. Cancer is, at its heart, cells dividing when they shouldn't be. And to divide, they need to replicate their DNA.
And to make DNA, they need a steady supply of all 4 DNTPs. And one of those, thymine, represents an Achilles heel.
Why thymine specifically?
Remember, RNA uses uracil, U. DNA uses thymine, T. T is basically just a methylated U. There is a specific step where a deoxyuracil nucleotide, DUMP, gets turned into a deoxythymine nucleotide, DTMP. The enzyme is thymidylate synthase.
And this is where the famous chemotherapy drug fluorouracil, or 5-FU, comes in.
5-FU is a suicide inhibitor. It's a brilliant molecular Trojan horse. It looks almost exactly like uracil to the enzyme. The enzyme grabs it, thinking it's a normal substrate, and starts the reaction. But once the reaction begins, that fluorine atom on the drug forms an unbreakable covalent bond with the enzyme.
It jams the machinery, it bricks the enzyme.
Permanently. The enzyme is dead. The cell cannot make thymine. No thymine means no DNA replication. The cancer cell tries to divide, hits this wall, and triggers apoptosis, programs cell death.
But it also explains the terrible side effects of chemo: hair loss, nausea, immune suppression.
Because those are also populations of rapidly dividing cells. Your hair follicles, your intestinal lining, your bone marrow cells, they're all constantly making new DNA. They are hammered by these drugs just as hard as the tumor is. Chemotherapy of this type is indiscriminate warfare on DNA synthesis.
Then there is another drug, methotrexate. It works differently.
Methotrexate targets the cofactor. Remember, to turn uracil into thymine, you need to add a methyl group, a single carbon. That methyl group is donated by folate, specifically a form called tetrahydrofolate.
And after it donates the methyl group, the folate has to be recycled or regenerated to be used again.
Right, and methotrexate inhibits the recycling enzyme, dihydrofolate reductase, or DHFR. So the cell quickly runs out of the active methyl donor form of folate.
Same result, different mechanism. No methyl donor, no thymine, no DNA.
And this highlights just how critical folate is, especially in pregnancy.
Spina bifida and other neural tube defects.
Yes. Those are essentially failures of DNA synthesis during the most rapid period of cell division in the developing embryo. If the mother is deficient in folate, the cells of the developing neural tube can't divide fast enough or correctly. Uracil might get incorporated into the DNA by mistake because there isn't enough thymine available.
And then the cell's own repair mechanisms come in, see the mistake, and try to cut the uracil out.
And in the process, they can cause double-strand breaks. The DNA shatters. The tissue fails to grow and close properly. That's why folate supplementation before and during pregnancy is one of the most important public health policies we have. It's pure biochemical prevention of a structural disaster.
It brings it all full circle back to the diet. We started with what we have to eat, essential amino acids, and we're ending with what we have to eat, vitamins like folate.
We are incredibly sophisticated biochemical engines, but we are completely dependent on the right fuel and the right parts from the outside to run the factory.
So let's try to wrap this up. We've gone from these simple carbon skeletons pulled from the Krebs cycle through the nitrogen funnel of glutamate and glutamine. We've dealt with the paradox of essential nutrients, watched the body build these incredibly complex rings for DNA, and then seen the tragic consequences when the regulation or the machinery fails.
It's a journey from the very simple to the profoundly complex.
What's your big takeaway? If you had to summarize the mission of this deep dive into enablism.
Well, I would go right back to your first metaphor, the conservative craftsman. The body is so incredibly efficient. It doesn't waste energy building things it can just get from the environment. It recycles absolutely everything it can via salvage pathways. And it regulates every single step with these intricate feedback loops to ensure perfect balance.
It's a system that seems designed for survival in a world of scarcity.
It is. And yet, for all its robustness, it walks this constant knife's edge. One missing vitamin, one mutated enzyme, and the whole elegant system can unravel into scurvy or PKU or Lesch-Nyhan.
That is the beauty and the terror of biochemistry, I guess. It works perfectly, almost invisibly, until it doesn't.
Exactly.
Here's a thought I want to leave everyone with. It comes from a line in Leninger. It mentions that the global supply of reactive, usable nitrogen is the ultimate limiting factor for life on Earth.
The nitrogen cycle.
Right. So, in a way, we are essentially just engines. We are engines designed to capture nitrogen from our food, reorganize it into our own specific proteins and nucleic acids, and then pass it on. We're just temporary custodians of nitrogen.
We are nitrogen processing units.
Exactly. So the next time you look at a steak or a bowl of lentils or a protein shake, don't just see food or calories. See it as a nitrogen harvest. You are gathering the fundamental raw material you need to rebuild yourself against the constant pull of entropy.
And if you really want to appreciate that complexity, don't just take our word for it. Open the books. Leninger on biosynthesis of amino acids and nucleotides, Harper's Illustrated Biochemistry on the same topics, plus the clinical correlations.
Look at the maps. Look at the structures. Trace the pathways. See the logic for yourself. Thanks for diving deep with us. We'll see you next time.
Goodbye, everyone.